I'm going to report back on the scientific basis for the OECD test guideline on in vivo toxicokinetic studies. So we had 2 sessions, and our aim was to gather some input on the gap analysis, on the study design requirements of in vivo toxicokinetic studies, and also to look into if there's any additional data that we missed and if we could prioritize the most important remaining data gaps. Next slide, please. So this is the agenda. First, we talked about the approach that we taken about the gap analysis, the identified gaps, and then we identified if there was any additional data. And the final point was prioritization of the gaps. Next, please. But first, before we dive into the results of the workshop, quickly, I would like to tell you a little bit about the approach. So toxicokinetics of nanoparticles are a little bit different from the toxicokinetics on molecular or ionic substances. Absorption is generally low, but once it is absorbed, it's usually quickly removed from the blood by phagocytotic cells, and it usually ends up in organs rich of these types of cells, so the liver and the spleen. From the liver and the spleen, and these type of secondary organs, they are usually very slowly eliminated. So this is one of the concerns of nanoparticles with respect to their toxicokinetics, that they accumulate in secondary organs. So this is what we want to address when we set up the test guideline. So we will be able to follow this tissue kinetics in primary, but also very important in secondary organs. So here I have on the screen a hypothetical graph of the concentration of the particles in the liver. And what we aim here is to see if we have enough in vivo toxicokinetic data available to roughly estimate how long we need and how high we need to expose, and how long we need to follow up after exposure. So how long the exposure period needs to be to follow either both the accumulation in secondary organs, but also the elimination phase. And then in able to do that, we need to know how many time points we need to measure in these organs and where we should put them. So what we'll use for this is kinetic modeling and, of course, all the data that is available, toxicokinetic data from in vivo studies. We focus on oral and inhalation, and we have decided to start with model substances, 2 model substances that are very slowly dissolving ones, the cerium dioxide and the titanium dioxide, and a moderately quickly dissolving one, silica. And please, next slide. So we discussed this approach and we got some general suggestions back because we focused on these particles, and it's particles, and there are lots of other materials, and they might be new materials as well. So there was a suggestion to broaden the applicability, not only to nanoparticles, but to other nanomaterials and even to larger scale particles. Another suggestion was the hypothetical curve might not be totally correct, because here after inhalation you see a lot of retention in the lung, so probably the liver concentration might accumulate for a far longer time after you stop the exposure period, so the peak needs to be shifted. Also a suggestion is to really define what we mean by very slowly, moderately quickly, and quickly dissolving nanoparticles, and how to measure this. And then not only the solution is important for determining the exposure and post-exposure periods needed, but also your limit of detection, your analyticals techniques, and the background concentration, of elements in the tissues, particle size, and administration mode. There was also a suggestion to not only look at these very slowly ones and the moderately ones, which we covered with our model particles, but also to include a quickly dissolving nanoparticle, for example, silver. And to use studies with radio-labeled nanoparticles as an example on how to set up this, the studies. Next slide, please. So besides this post-exposure and exposure and time frames, there are some additional aspects that we already identified that we need to address for the test guideline development. And these are on the left side. So that's what kind of toxicokinetic parameters are most important. How do you disperse and characterize your particles within the administration vehicle or matrix? The feeding status of the animals, administration mode, test species, strange age and sex of the animals, and also the use of in vitro data. Well, the experts in the groups, they came up with some additional aspects: animal welfare, blood kinetics, excretion via feces and urine. Sometimes you want additional organs than the ones we had identified, and it really depends on the purpose of your study. For example, if you know you have elements that can cause some toxicity, you want to include the organs where you expect this toxicity. To also take into account IV data besides the oral and inhalation, and to also not only look to the clearance from the tissues, but also from the whole body. Then, of course, we need to say something about number of animals per group to have to make sure you have enough power. And then there's this: if something dissolves within your body, a particle, it's also possible that new secondary particle formation takes place. So you have to take that into account. And something for oral exposure is that the volume of gavage might, even if it's below the ones that are in the ordinary test guidelines, it might influence your barrier function. Next slide, please. So thank you all for these additions. Then we moved on to the identified data caps. So we already discovered that there's not that many PBK models that are really general models, especially not for oral exposure. We're in need of some. And then we have in vivo toxicokinetic data for the very slowly dissolving particles, but for the moderately dissolving like silica, there's less data. And then we know the influence of several aspects that are mentioned, but sometimes it's not really clear what's the worst case situation. So that, having said that, that was automatically a reply from some experts that they came up with, well, for some of these aspects we do know. So for oral exposure, we know that fasted animals and juvenile animals is the most worst case situation. And for inhalation exposure, you know that if you disperse your particles very well, so there's less agglomeration and aggregation, that will be the worst case situation. There were some additional data gaps identified. So that's the retention of the particles within the delivery system. So that would be, for example, the tubes of your gavage. Well, for oral exposure, the dose that is actually available at the absorption surface. Then blood volume and volume distribution, specifically for nanoparticles, and the influence on size on the absorption and urinary excretion, distribution and elimination. That could be very useful. Next slide, please. So then with respect to the identification of additional data, we got some very nice suggestions for additional data that were not included in our gap analysis at first. So we'll have a look into that. They're from projects, unpublished ones, and REACH dossiers. Also some kinetic models we have to look into, and some data, as I told, that indicates what the worst case situation is in some cases. And there's some study underway that can give us more information on the retention of particles in delivery systems that we can use. With respect to prioritization of the most important data gaps, that was not really possible in our meeting. That's a tough question to answer from the top of your head, but we really gathered a lot of important issues. And from a regulatory point of view, it was mentioned that the insoluble ones, or the very poorly soluble ones, have priority. So yeah, we already took that into account by starting off with the very slowly dissolving particles as models. Next slide, please. So the implications really depend on the new data that we gather, the modeling that we do, and the interpretation of all the data all together. And then we have to prioritize the various aspects. And the next step will be to really update the gap analysis and to discuss this with the OECD Joint Expert Group. So there's a NanoHarmony webinar on the 16th of December in which we have some more extensive feedback or results presented. And of course, within NanoHarmony, we just keep on working on the data interpretation modeling. And we will deliver towards the OECD project a scientific basic for the study requirements. So thank you very much for your input during the last days. And if anybody has additional suggestions, I see a lot of things in the chat, but you're also very welcome to email them to me. Thank you very much.